So, for lecture number 2, we're going to begin to look at the contemporary evidence of human evolution. We're going to begin with DNA, which you should have some understanding of now. It's become an important part of sort of popular culture in the United States. It has even gone into K through 12 education today. So, my son was actually recently learning, and he's in middle school, some simple Mendelian genetics. And that's partially because of the rise in importance in DNA for such fields as criminal justice and medicine and so on. But we're going to be taking a kind of a distinct perspective. We're going to be looking at the importance of cellular and population genetics for evidence of human evolution. And by way of introduction, we can just say that what we know about DNA and what we know about genetics today does support an evolutionary understanding of species. It does support the idea of the formation of new species from ancestral ones. It also does support what we understand today about the relationships, the biological natural relationships between humans and other species. For example, 99% of human and chimpanzee DNA is the same. And so to understand the role of DNA as evidence for a theory of evolution, we sort of need to start thinking about evolution as changes in genes, changes in gene nucleotide frequencies, that is the patterns of genetic markers that manifest themselves as traits for the physical appearance of an individual organism. We can start to think about evolution really as a change in their frequencies through time. That when we have change at the genetic level, we have change at the species level. So, what do we really mean by genetics? Introduce this idea of DNA as an important evidence for human evolution, but what is the relationship of genetics here? Well, genetics really could be understood as the science of heredity. And heredity really is the way that traits, whether that be something like eye or hair color, range of different kinds of physical characteristics, it really is the way we study how those traits are passed on from one parent to offspring or from one generation to another generation. In fact, heredity is essential in understanding the way that traits are passed on and then the way that those traits change through time. A good starting point is the molecular and cell biological level. If you think about the human body, we have 100 trillion cells, right? And they all really have different structural and functional properties that are determined specifically by genes. They are determined by the genes located on the DNA in the cell nucleus, where those chromosome bundles are housed. Each cell, as you saw sort of in that human genome, has 2 pairs of 23 chromosomes, and 23 of those come from your mother and 23 come from your father. The totality of the human genome now has been mapped almost, it's really hard to believe, but almost 20 years ago, and we know of approximately 20,000 genes and a much larger number, of course, of nucleotides. Well, what are genes doing, right? Well, they are transcribing, if you will. The genome as a total is very, very big, 3 billion base pairs, but the code that genome represents provides instructions to the cell through the genes. A gene's DNA sequence is really transcribed into a molecule known as mRNA. And that process of transcription, the mRNA, is the way that DNA codes proteins that can create different kinds of cells, different kinds of cell structures, as well as different kinds of cell functions. So the DNA is in the nucleus of the cell, and it uses this mRNA to transmit those instructions, right, to the rest of the cell outside of the nucleus, that outer region that surrounds the nucleus, right, within the cell wall, sometimes referred to as cytoplasm. And it's in that area that the ribosomes, as you see there in the image, are going to read it, if you will, read that mRNA. That's a translation of the DNA code itself. And the ribosomes, these little machines in the cell, if you will, assemble the amino acids that are within that cell into the kinds of proteins that the DNA instructions provide. So it really is, as the cliche is, it's a building block of life. It's a building block of the human species. And there's two important processes to really think about that are relevant to our discussion about, genes or DNA as evidence of human evolution. And that is the process of mitosis and the process of meiosis. So when a cell divides in mitosis, the DNA must make a full copy of itself so that each new cell receives a full complement of code. If you think about the growth of the fetus, right, in the womb, if you think about the way that your skin repairs itself after a cut, you have this cell division that is a process, right, of organic life. And every time a cell divides, it must make a full copy of itself. It must have a full genetic code. And when the DNA divides, it makes full copies of itself, but it often does so with some degree of mutation, often which is harmless and has no really effects on the functionality or the structure of the organism. But it is an important source of genetic variation in code. If you think about that division of the code through time, the more divisions of the code that you have, the more reproduction of those instructions, the more potentiality we have for some kind of mutation to be included, as reproduction and replication, right, of the DNA is not perfect. So mitosis is kind of this nuclear DNA replication. And often we think about this focused on what are known as autosomes, the 22 chromosomes that are responsible for everything but determination of sex. So mitosis, right, is the bulk of this kind of replication for autosomes, those 22 chromosomes that do not determine sex. But meiosis, on the other hand, really is different, because meiosis is the process that we see in sex chromosomes. And what meiosis does is creates the gametes, or sex cells. And this is very important because while you also have the replication of the code through meiosis, this is really the method that we're going to have the DNA that is going to be transmitted across generations. So if you're thinking about the way that cells divide and DNA replicates in mitosis, let's say I get a cut and my body begins to repair that damage. And so cells begin to divide and replace, let's say, dead cells in that area. That is a source by which mutation is introduced into our genetic code. But that's going to be something that then is affecting an individual potentially. Can be longer term effects and so on, but generally speaking, if there's some mutations potentially in the cells that are going to be healing, right, a cut, That's very different than the kind of replication and potential changes that might occur when gametes or sex cells are created. Because it is through the gamete of the sex cells, right, that we're going to have uh, genetic information that's transferred across parents, right, to their offspring. And if we're thinking about the importance of DNA for understanding human evolution, it's meiosis then that becomes very, very important in understanding change through time. The way that this genetic code, which codes for the proteins and the building blocks of life, in the science of genetics, we understand that variation is introduced through division and replication in meiosis. So if you think about the 23 pairs of chromosomes, they're often termed homologous in the sense that they have sort of two sides, and through the sex cells of the gametes, both the male and the female provides half, right, of these pairs of chromosomes. And so through sexual reproduction, you have the female and male genetic data that gets combined. And what that means is that even though the DNA from each of the gametes, from each of the sex cells, is complete, right, the chromosomes are not. And so what you have in these bundled pairs of chromosomes is sometimes the same information for a trait, but sometimes we also have different information at the same location. That means in the same instructions on both of these chromosomes, but the determining aspects of the instructions, what they're actually telling a cell to do, is different. And this is something very important in sexual reproduction and meiosis because it introduces variation and variety. This combination through sexual reproduction allows for an allele, multiple variants, right, of one particular trait on the chromosome. You know, that's coming out of both the maternal and paternal components. Think about Mendel's experiments, for example. Mendel was a Czech scientist who really did help us understand how traits appear when these different genes are present. I mean, that was really the focus of somebody like Gregory Mendel's research. He conducted many heredity experiments using things like simple garden pea plants, and he concluded that there were traits that were always appearing. Mendel sort of inferred that there is this like unit, right, that was responsible for each characteristic and was passed on to offspring. And so rather than that blending, which was kind of the common understanding that somehow these things combined in offspring, each unit that a parent has actually continues. But there is a dominant form and there's a recessive form. And that is the Mendelian insight. And it's very important in understanding how variation, right, is contained within the genotype. From his research, he discovered a number of interesting things. This idea or principle of segregation, that each parent, the mother and the father, both contributed information, genetic information, but that they only have the ability to contribute sort of one allele. And the other, the second there, the independent assortment or principle of independent assortment, and the idea that the traits that are linked to different chromosomes, so remember there's the 23 pairs, traits that are linked to different chromosomes are inherited independently. So eye color and hair color, for example, are inherited independently from one another. But those cellular level kinds of discussion of genetics and the importance of DNA are only half the picture here of the role, right, of these in human evolution. When we're thinking about the ranges, right, of traits that are possible, and we're thinking about the different variants that are contained, sometimes it's simply two, and sometimes it's more than that, right? There's a polymorphism for a particular trait. There's a number of different possibilities there. And that number of different possibilities is really important in understanding the changes of forms through time, the frequency, right, of one form and its change through time. Essential here to understand is this not just at the level of two organisms who are reproducing sexually, but also the population as a whole. All of the possible allele variation for the population as a whole. Very, very important in understanding this change through time, the change, right, for a population through time. And there seem to be a number of different factors which we now understand in addition to the process of meiosis and how it produces variation at the allele level. And then at the trait level, the physical manifestation of the trait. This is what in the text we're referring to as the genotype versus the phenotype, right? The genetic structure of the individual, right? And then what is physically manifested on that basis of the genes. Well, when we're starting to think now about the changes, right, in the phenotypes and genotypes through time, we need to be considering, well, mutation. So we saw that in mitosis, but it can also exist in meiosis as well. It's an important way that variation is introduced. So you get the combination through the maternal and paternal contributions, but once they are combined and they begin to replicate, cells begin to divide and so on, you still have that introduction of mutation as the code is not perfectly reproduced each time. We also get things known as gene flow. So in our examples that we were giving of sort of isolated sexual reproduction, we are thinking about the kind of finite variance, right, that in sexual reproduction, a male individual organism might have, and the female individual organism may have. But in population genetics, that's not adequate to understand the population level frequencies of traits, the population level frequency of genotypes. And thinking about the single male and female and what kinds of variants are contained within them, but then scaling that up to think about, well, what are the total available variants there? Maybe myself, right, and my spouse, we have brown and green eyes. And maybe both of us have a kind of a dominant genotype for green eyes. And we even have it at the recessive level, greens at the recessive level. And my spouse has dominant brown eyes, but also in the recessive position is also a kind of a brown. And so we don't see blue and other varieties of eye color just represented in these two organisms. So thus we can't extrapolate logically to what's happening at the population level. If I was to divorce my spouse and I was to have another offspring with another genotype, it's going to introduce, right, new potentiality. They may have the genotype that contains information for blue eyes or other eye colors. And so this kind of gene flow is very important to understand changes through time. We have something called genetic drift. And genetic drift is, if you can think about it like this, is sort of non-random reproduction. And in the image, actually, we have a kind of a classic example of genetic drift, what is known as bottlenecking. And so In the left on this image, you see, a bottle filled with many different colored marbles. You have red, blue, green, and yellow. And the bottle's pretty full. But then when you try to empty that bottle into another one of these beakers here, you can see that only, a small amount of that substance, right, is going to be able to, by the shape, right, of that bottle, to make it itself into the beaker. You can't just turn this and all at once, all of the variety is going to make itself into that beaker. And a lot of these events, so genetic drift can be event-based, right? A lot of these events may be something like you have one population, but a small subgroup of that population migrates. And that small subgroup of the population that migrates will not be representative of the totality of the varieties. And so, as you can see here, you only have the blue and the yellow marbles. And as that population reproduces and grows, because the founder population, what's called founder effect, only had a fraction of the original population diversity, through that bottlenecking effect that happened, the end population looks very, very different, right? The traits that can be manifested there are different, even though its origin, right, was from one population. And that's because of that bottlenecking that occurred that then limits the kind of variation that can get passed. This is what we call genetic drift. And natural selection, as we've been talking about in the first lecture, a key component of the idea of evolution is that to understand the changes of traits through time, we have to understand the way that those traits are being selected for. All these forces result in genetic change over time. But natural selection means that the individual in a population with an advantageous gene is going to survive and reproduce in greater numbers. That's going to cause those alleles to increase in the population. And so we have to remember that there is no apparent evolution if there is no change in frequency of genes such as we're talking about here. And this is that idea behind the Hardy-Weinberg equilibrium. The conditions for the Hardy-Weinberg equilibrium are theoretical. The idea of no change, the conditions that are required, right, for a population to have no evolution, to be not undergoing evolution. Sort of in this sense, that cliche that change is the norm is quite accurate. A good example is that we now know that human mitochondrial DNA has a clear nested structure of 7 or 8 haplotypes, or different groups of nucleotides that are clearly identifiable. This means all living humans descend in an unbroken line through their mothers to this one single woman. Using what we understand about how mutations accumulate in mtDNA and looking at the patterns of that diversity, we estimate that that particular woman whose mtDNA now defines the human species lived approximately 140 to 200,000 years ago in East Africa. And that's true as well for if we're thinking about humans and the relationship between one population and another, but also the evidence, right, for humans and their relationship to other species. So in 2005, we had the first true detailed comparison of human and chimpanzee DNA. And that's where we were able to ascertain that we're 99% identical in terms of our DNA with chimps, and just a little less relation with bonobos, which most people could not tell the difference between a chimp and a bonobo, but they are genetically distinct, and we're actually a little bit closer to chimps than we are to the bonobos. This is why genetics is an important form of evidence for human evolution today. We can actually see and calculate how many changes have occurred on the DNA between humans and chimps, so that there has been calculated roughly about 35 different nucleotide changes, and then 5 million either taking away of parts of the code that was shared with chimps, right, or adding new parts of code that is not there with the chimp. And so that's even less than if you were to compare the genotype of a mouse and a rat. In fact, mice and rats are further in terms of their genetic distance than humans and chimps. And in fact, humans and chimps, it's, as you can see here, it's like chromosome 8, and the sex chromosome, right, is areas where you see a heightened amount of variation. It's not even the fact that that variation is across all of the chromosomes. You have huge areas of these chromosomes in chimps and humans that are exactly the same. And then you have areas, right, where you had changes, you may have had some insertion, you may have longer stretches of code that was added in our own evolutionary history. But by far, the majority of that genome, just like we could say for all human populations that the majority of our genome is shared, is uniform. You can track differences between populations. This is something like Ancestry, .com and so on, where we know the haplotypes that are out there and what populations they're associated with. But that doesn't mean that the majority of the DNA of humans is shared and, in fact, is ancestral to us. And when we're looking at the chimps and humans, it is precisely the same way. Actually, the bulk of the genes for humans and chimps is ancestral. That is, it had developed before the development of humans or chimps. And we could say that life on Earth also has that kind of nested hierarchy as well. Something that I have in the DNA code, the gene code documentary in this unit of the course. So to kind of conclude here, one contemporary form of evidence for human evolution is DNA. On the one hand, cellular genetics really helps us understand biological relationships between one human and another, particularly the principles of Mendelian genetics, sexual reproduction, and how variation is introduced into DNA. And on the other hand, the population genetics, really understanding higher order relationships, right, between human groups, populations, and the higher order relationships, right, between other kinds of taxonomic groupings, let's say within the primates or between primates, right, and other species.